Welcome to a brand new series of our explainer. Okay, so we've successfully made our way through glycolysis in the cytosol, but now it's time for the big leagues. We're moving right into the mitochondria, the absolute power plant of the cell. Today, we're zooming in on the crucial bridge between our last topic and this one, the Pyruvate Dehydrogenase Complex, or PDHC for short.
All right, here's our roadmap for today. We'll cover the journey to the matrix, the PDHCl relay race, the coenzyme toolbox, clinical correlates and stakes, and finally, wrap it up with some exam prep and a recap. Let's get to it.
First up, our journey to the matrix. So, in our last series, we left our main character, pyruvate, just hanging out in the cytosol. But here's the catch. The enzymes for the TCA cycle, they're all tucked away inside the mitochondrial matrix. And since pyruvate is a polar molecule, it can't just casually float through the inner mitochondrial membrane on its own. No, it relies on a very specific transport protein to cross over into the mitochondrion.
You can kind of think of this transport protein like the security gate at a massive stadium. If pyruvate doesn't get through this facilitated first step, it absolutely doesn't get to see the main show, which of course is the TCA cycle. It's an exclusive VIP area, and this gate is literally the only way in.
Moving right along to the PDHC relay race. All right, once pyruvate finally makes its grand entrance into the matrix, it meets the star of today's explainer, the Pyruvate Dehydrogenase Complex. Now, a crucial thing to grasp here is that this isn't just some solitary, floating enzyme. The PDHC is this massive multi-enzyme aggregate made up of multiple copies of 3 distinct enzymes, working in absolute perfect harmony. We call them E1, E2, and E3.
And by physically sticking these 3 enzymes together, that's pyruvate decarboxylase, dihydrolipoyl transacetylase, and dihydrolipoyl dehydrogenase, the cell ensures total efficiency. The chemical intermediates are handed off perfectly, almost like a baton in a rapid-fire relay race. They move in sequence without ever releasing those precious intermediates into the wild, which prevents any loss.
So what's the overall goal of this whole metabolic relay race? Basically, the PDHC takes that 3-carbon pyruvate and transforms it into a 2-carbon acetyl-CoA. During this process, carbon dioxide is released and boom, we successfully harvest our very first mitochondrial NADH. It is just a beautifully coordinated maneuver.
Next up, let's look at the coenzyme toolbox.
To pull off this wild biochemical sequence, our massive 3-enzyme machine needs a very specific set of tools. They act as carriers or catchers for the molecules as they get passed along. Now, if you're prepping for exams, you absolutely must memorize these 5 coenzymes and the specific subunit they pair with. We use this classic mnemonic, tender, loving care for Nancy. That stands for TPP, derived from vitamin B1, for E1, Lipoic Acid for E2, CO from vitamin B5, also for E2, FAD from vitamin B2 for E3, and NAD+ from vitamin B3 for E3. Tender, loving care for Nancy. Seriously, burn that into your memory.
But not all the tools in this toolbox act the exact same way. TPP, lipoic acid, and FAD are what we call prosthetic groups, meaning they are tightly bound to their enzymes and stay there permanently. On the flip side, CoA and NAD+ are mobile coenzymes. They come and go, acting more like flexible delivery trucks whenever they're needed.
All right, on to clinical correlates and stakes.
Now that we know how this intricate machine works under ideal conditions, we really have to look at the severe consequences when things break down. We have to ask a pretty terrifying question. What happens when the brain's power plant goes dark? Because our brain relies almost exclusively on the TCA cycle for its massive, non-stop ATP demands. A shutdown of the PDHC due to vitamin deficiencies creates an immediate, catastrophic energy crisis.
For instance, without the T in our tender loving care, meaning thiamine or vitamin B1, the brain literally starves. This leads directly to Wernicke-Korsakoff Syndrome, which is a highly serious condition involving encephalopathy and psychosis. Actually, this is frequently seen in cases of chronic alcohol abuse, where poor nutrition leads to a critical lack of thiamine, just completely shutting down the PDHC.
And arsenic poisoning raises the stakes even higher. Trivalent arsenic works in a very similar, nasty way by specifically targeting and forming a stable complex with the thiol or SH groups of lipoic acid. This renders that vital E2 coenzyme completely unavailable, abruptly halting the PDHC. pyruvate and lactate rapidly build up, causing severe neurologic disturbances and, tragically, often death.
Finally, let's hit our exam prep and recap. Okay, let's lock in everything we've covered today with a rapid-fire active recall session. Get ready to test yourself. First recall question. Where specifically in the cell is the PDHC located? Take a quick second and think back to our map of the cell from the very beginning.
Got it? The answer is the mitochondrial matrix. And remember, this is exactly why our polar pyruvate needed that facilitated transport protein, that stadium security gate, to cross the inner mitochondrial membrane in the first place. All right, now for an application question. A patient presents with confusion and ataxia linked to chronic alcohol abuse. Which specific PDHC coenzyme is likely deficient? Think back to our clinical stakes and that handy mnemonic we used.
The answer is TPP, or thiamine pyrophosphate. Its precursor is vitamin B1, which gets heavily depleted in cases of chronic alcohol abuse and poor nutrition. This deficiency directly disables the E1 subunit, leading right to Wernicke-Korsakoff Syndrome.
Finally, a conceptual question. Why does a thiamine deficiency affect the brain so much more severely than other tissues in the body? Let's connect the microscopic biochemistry to the macroscopic reality of our organs.
Well, here's why. The brain relies almost entirely on the TCA cycle for its massive energy needs, and the PDHC is the absolute only bridge to supply the necessary acetyl-CoA from glucose. While other tissues might lean on alternative pathways like fatty acid oxidation, the brain is just incredibly greedy for TCA cycle energy. If the PDHC bridge collapses, the brain starves almost immediately.
I'll leave you with this final thought. The PDHC is the ultimate metabolic bridge. If you don't get through here, you don't get to see the main show. In summary, this incredible 3-enzyme machine and its 5 vital coenzymes represent the absolute threshold for aerobic life. So, if this bridge is absolutely essential for our survival, what incredible mechanisms regulate it when our body is resting versus when we're sprinting? That is a fascinating question for our next explainer. Keep learning, and I'll see you next time.
Hi, everyone. So, in our last explainer, we met the massive 3-enzyme machine known as the PDHC, right? And we talked about the 5 vitamin-derived coenzymes it desperately needs to work. Well, today, we're popping the hood. We're going to trace exactly how a single molecule of pyruvate is decapped, oxidized, and then loaded onto coenzyme A. After that, we'll look at the master control switches, the kinases and phosphatases that ultimately decide when this engine should be revving at full speed. Let's get into the mechanism and regulation of the PDHC.
Here is our quick roadmap for today. We'll start by looking under the hood of the PDHC, move on to the molecular relay race, examine the covalent switch, dive into allosteric control signals, and finally, do a little exam prep.
Okay, Section 1. Under the hood of PDHC.
You probably already know that glycolysis happens out in the cell's cytosol, producing pyruvate. But the TCA cycle, where we extract the real bulk of our energy, happens deep inside the mitochondria. So pyruvate actually has to be transported across the inner mitochondrial membrane and into the matrix. That matrix is the factory floor. It's where pyruvate arrives, ready to meet the Pyruvate Dehydrogenase Complex and be transformed into acetyl-CoA.
Now, the PDHC isn't just one simple solitary enzyme. It's this massive protein aggregate made of multiple copies of 3 distinct components, E1, E2, and E3. The absolutely crucial point here is their physical association. Because these 3 enzymes are literally glued together, the reactions are linked sequentially. The intermediates are never released to just float away into the cellular abyss. Instead, they are passed directly from one enzyme subunit to the exact next one, making this an incredibly efficient assembly line.
Moving right along to Section 2, the molecular relay race.
Think of the PDHC as a highly coordinated molecular relay race. There are 5 distinct steps passing batons of carbons and electrons. We start with decarboxylation at E1, move to oxidation and transfer, and then form our final product, acetyl-CoA, at E2. Then, steps 4 and 5 at E3 are really just all about cleaning up and resetting the machine so it's ready for the next run.
Let's break down this 5-step sequence. First up is decarboxylation. Pyruvate meets the E1 subunit pyruvate decarboxylase. Using the coenzyme TPP, the enzyme literally snaps off the top carbon of pyruvate as CO2. What's left behind is a 2-carbon hydroxyethyl group still stuck to that TPP. Second, we have oxidation and transfer. The E2 subunit dihydrolipoyl transacetylase has this amazing swinging arm made of lipoic acid. This arm reaches over, grabs the 2-carbon fragment from TPP, and oxidizes it into an acetyl group. This acetyl group is now covalently bound to the sulfur atom of the lipoic acid. Third is the formation of Acetyl-CoA. That swinging arm physically moves the acetyl group over to a waiting coenzyme A molecule. This creates Acetyl-CoA, the main product we need for the TCA cycle. But notice that after this handoff, the lipoic acid is left in a reduced state, meaning it's holding on to 2 hydrogens. So, fourth is the reoxidation of lipoic acid. We have to reset the machine. The E3 subunit, dihydrolipoyl dehydrogenase, uses FAD to strip those hydrogens off the reduced lipoic acid, turning it back into its active oxidized form. This reduces FAD to FADH2. And finally, step 5, regeneration of FAD. The FADH2 on the E3 subunit is reoxidized back to FAD by handing its electrons to NAD+, which then leaves as NAD+, plus a proton. And with that, the baton has officially crossed the finish line.
So, when the dust settles on all those molecular acrobatics, this entire sequence ensures that for every single pyruvate entering the PDHC, we get exactly 3 things. 1 acetyl-CoA to feed the TCA cycle, 1 CO2 exhaled as waste, and 1 NADH packed with high-energy electrons.
That brings us to Section 3, the covalent switch.
Here's the thing. Because the PDHC reaction is completely irreversible, meaning once you make acetyl-CoA, you absolutely cannot go back to pyruvate, the cell regulates this complex like a strict military checkpoint. The primary way it achieves this is through covalent modification of the E1 subunit. The PDHC actually has 2 helper enzymes built right into it. Think of PDH kinase as the off switch. It adds a phosphate to the E1 complex, essentially making it inactive. If PDH kinase is active, pyruvate cannot enter the TCA cycle. On the flip side, we have PDH phosphatase. This is our on switch. It uses water to snap that phosphate right off, activating the complex once again.
Here's a quick memory trick for your exams. I want you to remember this simple rule. Phosphorylation equals pyrolysis. When the PDH kinase adds that bulky phosphate group onto the E1 subunit, the enzyme is paralyzed. It's inactive. The whole factory shuts down. Section 4. Allosteric control signals.
So we know about the on and off switches, but what tells the kinase and phosphatase to actually do their jobs? Well, the cell uses allosteric signals, essentially chemical messengers, based on its immediate energy needs. If the cell has high levels of ATP, acetyl-CoA, and NADH, it activates the off switch. These high-energy signals tell the kinase to shut down the PDHC, because hey, the cell already has plenty of fuel. But what if there's a massive flood of incoming substrate? Pyruvate is actually a potent inhibitor of the kinase. It blocks the off switch, ensuring the PDHC stays active to process all that incoming sugar. Finally, what activates the phosphatase, our on switch? The answer is calcium. And that leads to our second memory aid for today. Calcium is the gym teacher. This is incredibly critical in skeletal muscle. When you suddenly start running, your muscles release a flood of calcium to trigger muscle contraction. But as soon as that calcium is present, it acts literally like a gym teacher blowing a whistle. That exact same calcium rushes over to the PDHC and tells the phosphatase to wake up. The phosphate is removed, the PDHC snaps out of its paralysis, and you immediately start producing the massive amounts of energy you need for that run. It's a beautifully elegant system.
Finally, Section 5, Exam Prep. Let's lock this in. Let's tackle a classic conceptual question. Why is the physical association of E1, E2, and E3 into a complex metabolically advantageous? Well, as we saw on the factory floor, it's all about efficiency. By physically linking these enzymes, the intermediates don't have to aimlessly diffuse through the cell to find the next enzyme. They're handed off directly via that swinging lipoic acid arm. This totally prevents the loss of intermediates and dramatically speeds up the overall reaction.
Next up, explain why high levels of acetyl-CoA lead to the inactivation of the PDHC. Remember our allosteric signals. Acetyl-CoA is the end product here. If you have a ton of it, it means the cell's energy needs are already met. This abundance of fuel allosterically activates PDH kinase, which then slaps a phosphate onto E1, shifting it straight into pyrolysis mode to stop any unnecessary overproduction.
Another common one, how does the regulation of PDHC by calcium link muscle contraction with energy production? This goes right back to our gym teacher analogy. The physiological trigger for a muscle to contract
is the release of intracellular calcium. By having that very same calcium also activate PDH phosphatase, the cell perfectly synchronizes the demand for energy, the muscle contraction, with the supply of energy by instantly turning the PDHC on.
Now here's a tricky one. Why is the E3 subunit necessary even though acetyl-CoA is already formed back in the E2 step? The crucial point is that enzymes absolutely must return to their original state to catalyze another reaction. While E2 does make the final product, its swinging lipoic acid arm is left holding 2 hydrogen atoms. It's reduced. E3 is necessary for resetting the machine, stripping those hydrogens off, so the arm is oxidized and ready to catch the very next pyruvate.
And finally, contrast the role of FAD in the PDHC with its role as a prosthetic group in other enzymes. Unlike free-floating coenzymes like NAD+ that kind of just come and go, FAD is tightly, permanently bound to the E3 subunit. It acts as a fixed middleman, accepting electrons from lipoic acid and then waiting patiently for a passing NAD+ to take them away. Honestly, it makes you wonder, how many other enzymes out there rely on these hidden, tightly bound molecular middlemen to pull off these incredible feats of chemistry? Keep that in the back of your mind as you continue exploring metabolic pathways. Thanks for joining me on this explainer, and keep learning.
Hi everyone, and welcome to The Explainer. We've spent the last few lessons admiring the incredible complexity and regulation of the pyruvate dehydrogenase complex. But what happens when this massive molecular machine breaks down? Today, we're looking at the clinical side of the PDHC. We'll explore why a tiny genetic mutation can lead to devastating neurological diseases like Leigh syndrome, and how a toxic element like arsenic can physically handcuff your enzymes. Let's dive into the clinical correlates of the PDHC. Here's our quick roadmap for today. We'll start with when the PDHC breaks, then move on to congenital lactic acidosis, understanding Leigh syndrome, the threat of arsenic, and we'll wrap it all up with some exam prep and review. All right, section 1, when the PDHC breaks. Let's get right into it. So I want you to imagine for a second the absolute metabolic chaos of sabotaging the cell's most critical energy gateway. The PDHC is literally the bridge between glycolysis and the TCA cycle. If you, say, blow up that bridge, well, where does all that cellular traffic go? In a normal, totally healthy state, pyruvate just seamlessly passes from the cytosol right through the inner mitochondrial membrane and into the matrix. Once it's there, it's oxidized by the PDHC into acetyl-CoA, creating NADH and releasing CO2. I mean, this is aerobic respiration working exactly as intended, right? But what if we block that transport? Or what if we break the complex that's waiting on the other side? That brings us to section 2, congenital lactic acidosis. The first major condition we really need to discuss is congenital lactic acidosis. Now, while there are a lot of different causes of lactic acidosis out there, the most common biochemical cause is actually a deficiency in the alpha subunits of the tetrameric E1 component of the PDHC. Remember, E1 is the gateway. It's the part that performs the initial decarboxylation. So if E1 isn't working, the cell has a major traffic jam on its hands. Pyruvate literally cannot enter the mitochondria to become acetyl-CoA. The main road is totally blocked off. So what happens with all that backed up traffic? Well, instead of moving forward, pyruvate gets shunted to lactate via the lactate dehydrogenase reaction we studied back in our first course. Basically, NADH is oxidized to NAD+ as pyruvate is reduced to L-lactate. And that massive buildup of lactate in the blood, that is exactly what causes severe acidosis. Now, this creates a massive problem for the brain. Your brain is an absolute energy hog. It relies almost entirely on the TCA cycle for its ATP. When the PDHC fails, the brain is double hit. It loses its primary energy source and becomes severely damaged by the resulting acidosis. Symptoms of this deficiency are quite variable, but they often include things like neurodegeneration, muscle spasticity, and in severe neonatal forms, early death. While there's no actual cure, doctors often try restricting dietary carbohydrates and providing thiamine supplements. Why? To help squeeze every last bit of activity out of what little enzyme actually remains. Okay, moving on to section 3, understanding Leigh syndrome. Closely related to this is Leigh syndrome, which is also known by the rather intimidating clinical name, subacute necrotizing encephalomyelopathy. Let's break that down. This is a rare and progressive neurodegenerative disorder. It isn't always caused by a PDHC defect specifically, but it is always caused by a failure in mitochondrial ATP production. This catastrophic energy failure can actually result from mutations in the genes that encode the PDHC, components of the electron transport chain, or even ATP synthase itself. Because the brain is so incredibly sensitive to energy fluctuations, these defects lead to the necrotizing or tissue death that is characteristic of the disease. Basically, without ATP, the brain's cellular machinery just shuts down. Let's shift gears for Section 4, the threat of arsenic. Now, let's talk about a classic toxicology topic, arsenic poisoning. In our first course, we saw how pentavalent arsenic, or arsenate, mimics phosphate. But today, we're looking at arsenite, the trivalent form of arsenic, and these two operate through entirely different mechanisms of sabotage. So arsenite has a very specific, dangerous affinity for the thiol, or sulfur groups of lipoic acid. Remember our swinging arm from Lesson 2? That flexible arm is made of lipoic acid. Arsenite comes in and forms a stable, permanent complex with those thiol groups, effectively freezing the arm. It basically slaps a pair of physical handcuffs right on the enzyme. Because lipoic acid is absolutely essential for E2, the entire PDHC shuts down. But it doesn't stop there. Arsenite is a serial saboteur. It also inhibits other complexes that require lipoic acid, such as the alpha-ketoglutarate dehydrogenase complex, which we'll see later in the TCA cycle, and the branched-chain alpha-keto acid dehydrogenase. Just like with genetic PDHC deficiency, this leads to a massive accumulation of pyruvate and lactate, causing severe neurologic disturbances and, eventually, death. Finally, Section 5, Exam Prep and Review. To remember the difference between the two types of arsenic, I want you to memorize this. Arsenate, the pentavalent one, acts like phosphate. Arsenite, the trivalent one, bites the lipoate, meaning lipoic acid. It's a silly little rhyme, but trust me, it's absolutely crucial for test day. And for Lee syndrome, just remember, Lee leads to low energy because it hits the PDHC, ETC, or ATP synthase. It's a perfect way to anchor the low energy state to the syndrome's name. Let's test your active recall. Why does a deficiency in the E1 subunit specifically lead to an increase in blood lactate levels? Well, think back to that traffic jam we discussed earlier. Because E1 is broken, pyruvate can't be converted to acetyl-CoA. So the enzyme lactate dehydrogenase kicks into overdrive, converting that backed up pyruvate into lactate, which causes the lactic acidosis. Okay, next question. A patient is exposed to a toxin that specifically binds to thiol groups. Which coenzyme of the PDHC is the primary target? If you remember our handcuff metaphor, you know this is arsenites, and it's targeting the lipoic acid swinging arm on the E2 subunit. Let's try a bit of an application question. Why might a ketogenic, meaning high-fat, low-carb, diet be prescribed for a patient with PDHC deficiency? Think about it. Carbs break down into glucose, which becomes pyruvate. If the PDHC is broken, more carbs just mean more lactic acidosis. By switching to a high-fat diet, the body actually bypasses the PDHC and generates acetyl-CoA directly from fats, providing an alternative fuel source without totally overloading that broken pyruvate gateway. We've seen today just how incredibly fragile our cellular energy production can be. A single genetic typo in the E1 subunit or a microscopic dose of trivalent arsenic is quite literally enough to bring the entire system crashing down. So it leaves us with a pretty provocative thought. If energy production can be hijacked so easily by just one faulty subunit or toxic element, what other metabolic gateways might be vulnerable in our cells? Keep that in mind as we continue our biochemical journey. Thanks so much for joining me on this explainer, and I'll see you next time.